traits are also known to respond to temperature, including anthocyanin deposition and alternative oxidase concentration in the flowers, as well as the timing of flowering onset and spike production, the length of these scapes. As you can see, they're very long in warm environments and shorter in cool environments, as well as the percentage of viable pollen and floral pubescence, which are small microscopic hairs that I'll show you later, and some vegetative traits, including leaf length and leaf angle, also respond to temperature. So on to my AFLP study, and this study is designed to answer this question: does neutral divergence contribute to thermal climates? So in this study, I'll test the following 4 hypotheses: isolation by distance, glacial refugia on southern peninsulas with subsequent migration northward, that patterns of plasticity can be explained by neutral divergence, and that temperature and duration of the reproductive season best explain these patterns of plasticity along thermal climates. So to test these hypotheses, I'm using 14 populations from southern Europe, shown here. And these span a latitudinal range from southern Italy up to Germany. And these 4 populations in the center represent an altitudinal climb with FRG at the bottom of the French Alps and moving up that mountainside to FRR at the top of the French Alps. Also on this map, I've shown the maximum extent of glaciation during the Pleistocene glaciation maximum about 20,000 years ago in shaded blue. And this dashed line represents the southern limit of permafrost. The reason I put these on there is because many species throughout Europe have been found to have survived the last glaciation in these southern peninsulas, and after the glaciers receded, were able to then repopulate northern Europe. So I want to see if Plantago follows those same patterns. So to get my data for this amplified fragment length polymorphism analysis, I first extracted DNA from each individual and digested it using 2 restriction enzymes, EcoR1 and MSE1. Those fragments were then selectively amplified using PCR with 8' pair and 2 di combinations. And then I evaluated fragment size on a capillary sequencer and scored DNA fragments in gene marker. So here I'll show you how I did that. Signal intensities on the y-axis and length of those nucleotide fragments on the x-axis. And here I'm showing one individual from the FRM population and a separate individual from the FRG population. So we can see that at the fragment length of 240 nucleotides, both individuals show a peak here. And so they were both scored as positive, having a peak at that length. However, at 243 nucleotides, there's a peak at the FRG individual, but no corresponding peak in this individual, so that represents a polymorphism. And it's these differences between individuals that I'm using to calculate my variables to look at genetic diversity, differentiation, and similarities. So all of my variables together, I'm using location of population origin as my independent variable, and genetic diversity measured as mean heterozygosity, genetic differentiation between populations, and this value, or this statistic that falls between the values of 0 and 1, with 0 representing no differences between those populations, essentially one large population, and 1 representing no similarities or completely separate populations. I'm also using Bayesian clustering of individuals in program structure. And in this program, I put all of my individuals into the computer and have the computer sort them into 2 groups based on similarity. Then I'll reset the analysis and have the computer sort them into 3 groups and 4 groups and so on. So I can see which individuals separate out as similarity increases. And the last variable is phenotypic differentiation between populations, in which I'm using the phenotypic variance to calculate. So to test my first 2 hypotheses, I'll be using Mantell tests to examine correlations between genetic differentiation genetic diversity, and physical distance. And I'll use that Bayesian clustering program to examine patterns of similarity and specifically answer the questions, are the populations in the Iberian and Italian peninsulas separated when it's k equals 2? And then which populations separate out as I increase the value of k? And k is the number that I specify the computer to sort them into. So if I sort them into 2, 3, 4. So these populations predict that differentiation will be positively correlated with distance between the populations. Diversity should be highest in the oldest populations or populations from multiple ancestries. And populations should cluster into their ancestral populations at k equals 2. So this is where I am so far in addressing these hypotheses. This is a figure of mean heterozygosity, my value of genetic diversity, plus or minus 95% confidence intervals. And I have increasing diversity shown in warmer colors with lower diversity shown in cooler colors. So the first thing to notice is that there's not really a clear pattern of south to north that isn't showing up in this data. What we can see is that this IB population has the highest diversity, and that's about it from there. To calculate differentiation, what I first did was some preliminary analyses between all populations, and those suggested that I could group these populations based on regions, because all of these populations were very similar with each other. I then did a regional differentiation comparison, and here I'm showing populations or regions connected by lines. These are all very similar populations because they have an FST value less than 0.1. So here we can see that IB population that had the highest diversity may have some ancestry from this German, these Italian, and these Spanish populations. And from this it suggests that individuals from this Iberian Peninsula may have gone to populate these French populations. Here's the output from the structure analysis, and one of these lines represents one individual in the analysis. So here I'm told the computer to separate it into two groups based on similarity. And for example, this small black line suggests that that individual has about 80% of its genome from the black group, or 80% from the yellow group and the black 20% from the black group. So what we can see here is that these Italian populations are the black ones and the Spanish populations are yellow. So it looks like the populations on these two peninsulas were separated at k equals 2. When we go into k equals 3, now we can see that the Italians remain very distinct from the other populations. That suggests that perhaps there were these Italian and French Alps were served as a barrier to migration northward. To answer my third hypothesis, patterns of plasticity can be explained by neutral genetic divergence. I'll examine the correlations between phenotypic plasticity, genetic differentiation, diversity, and physical distance. And this hypothesis predicts that I'll find a positive relationship between these variables. In addition, I'll examine phenotypic differentiation and genetic differentiation to examine the relationship between those variables. And if I find that they are equal and fall on this line of equality, genetic drift will be sufficient to explain patterns of plasticity. However, if phenotypic differences are greater than genetic differences, that will be in support of divergent selection causing the patterns of plasticity, and the opposite trend will support stabilizing selection. My fourth hypothesis is that temperature and duration of the reproductive season best explain plasticity. And to test this hypothesis, I'll use multiple regression, so I can control for genetic and physical distances while I test the effect of temperature, duration of the reproductive season, and precipitation on patterns of floral reflectance plasticity.